For our final presentation, we'll turn to Dr. Terina Martinez, Executive Director of the Duchenne Regulatory Science Consortium and the Critical Path to Therapeutics for the Ataxias at Critical Path Institute, to talk about neurofilament light chain and its use as a biomarker in neurological disorders. Dr. Martinez.
Thank you, Dr. Lee, for the introduction, and thank you, everyone who's organized this wonderful workshop today. It is my benefit and pleasure to come after so many other excellent speakers who've done such an excellent job providing the foundational groundwork for the various considerations and applications for biomarkers, and those really key questions that we need to ask as we're trying to develop biomarkers for clinical trials at all levels, for surrogate endpoints, for prognostics, et cetera. So I just really want to thank everyone for staying with us. I am Terina Martinez with the Critical Path Institute, and my goal here is to provide kind of like a toggled landscape of looking at a candidate biomarker that has different position and development process in different neurological diseases, and try to understand what we can learn from the mechanism of action of this biomarker and what needs to be considered to develop it in different individual neurological disease applications. So next slide, please. I do not have a disclosure slide because the Critical Path Institute is a nonprofit agency. We are focused on advancing science through cross-disciplinary collaboration. The Critical Path Institute was founded in 2005, located in Tucson, Arizona. It's an independent, not-for-profit organization really focused on a public-private partnership model to convene scientists across sectors, including scientists from regulatory agencies, drug development industry, academia, clinician researchers, all to come together to collaborate and improve the drug development and regulatory process for medical products. The goal is to leverage this public-private partnership and the consortium model to create consensus, define paths, and improve health. Next slide, please. Within the Critical Path Institute, we actually have many different disease area focuses for our various consortia. Some of our consortia are disease-specific and solution agnostic. Others are solution-specific but disease agnostic. So an example of some of our disease-specific consortia are within our neuroscience program. That includes our Critical Path for Alzheimer's disease, Critical Path for Parkinson's, the Critical Path to Therapeutics for the Ataxias, Duchenne Regulatory Science Consortium, and the Huntington's Disease Regulatory Science Consortium. But we are just the neuroscience side of the institute. We also have a consortia based on sickle cell disease, polycystic kidney disease, and even disease agnostic ones like clinical outcome assessments. So today, though, I'm going to go through this thematic outline looking at neurofilament light chain, which I'll abbreviate as NFL, and specifically the molecular mechanism and biological significance of it as a biomarker and what this means in different neurological diseases. I'll consider its potential across diverse diseases and provide a little bit of a deeper dive, obviously not a very deep one due to time, for multiple sclerosis. I will address current challenges and knowledge gaps for developing NFL as a clinical trial-ready biomarker, and also discuss best practices for advancing neurofilament light as a fluid biomarker for neurological disease in a manner that is maximized for regulatory success. I'm also the last speaker in this session, so I will lead us into the panel discussion. Next slide, please. So this is just a brief overview on NFL biology and mechanism, just so that we can understand its position and why it actually has potential application across diverse neurological diseases. So neurofilaments are an abundant neuronal scaffolding protein. There are many different subunits within this scaffolding. Neurofilament light chain is one of the subunits, and it is the one that is in favor for most biomarker applications. I want to point out that it is highly specific for neuroaxonal damage, but that importantly, while it's very specific for this damage, it is agnostic to the primary neuronal damage trigger. What's really, I think, advantageous to NFL is that it has very good potential as a plasma biomarker for neurological disease, which is kind of a moonshot for neurological disease, because of course, having to do lumbar punctures for CSF fluid sampling is fairly invasive. What's great about NFL is that it is detectable in both compartments, CSF and blood, and first generation assays to measure We're certainly sensitive to detect it in CSF, but it's really been the development of third generation and fourth generation assays. The one that's really the most advanced at this point is the Simoa assay, is able to detect it at really good levels and sensitivity in blood. So while the overall levels in blood are lower than in CSF, there's good linearity between the two compartments. So you can reasonably interpret observations of NFL in blood as a snapshot of neuronal axonal damage that's occurring within the brain. Next slide, please. In this point, I want to contextualize the mechanisms of NFL with divergent and conserved mechanisms in neurological disease. So there's selective neuronal vulnerability and brain region specificity and progressive pathology underlying the complex etiology of neurodegenerative diseases. That's depicted in this schematic here with the different colors highlighting different nuclei and brain regions that are associated with very specific neuronal populations that have specialized tasks and neurotransmitter components. And the insult to any one of these very discrete brain regions, and therefore the effect on the neuronal population and neurotransmitter affected, will then manifest in diverse clinical manifestations that really do specifically reflect that brain region and this specific population affected. You can also see from the schematic that there is a really unique sub-architectural organization to these different neurons. Whether we're talking about medium spiny neurons in the striatum in Huntington's disease, which are highly not quite as arborealized as dopaminergic neurons, but do have a lot of axonal extensions, or even the motor neurons that are typical in ALS. These neurons have different structures, and the amount of their axonal subunits and the structural subunits of the neurofilaments are going to be different across them. And so while we're making the case that neurofilament is a sensitive measure of neuroaxonal damage, we're absolutely acknowledging that it is not disease specific, but it still has really good potential to be leveraged as a biomarker across diverse neurodegenerative disease. Next slide, please. This is a schematic that's really pulling together kind of the broad conceptual roadmap that we're beginning to understand more as many different researchers globally are providing the results and evidence to support the function of neurofilament light in various neurological diseases. We know with most neurodegenerative diseases that there is an insidious onset of disease symptoms. Often these diseases occur on the background of aging. And for example, in cases like Alzheimer's disease or age-related diseases like Parkinson's disease, there's several decades without symptomology, but then the symptoms do manifest. So this is a schematic looking at serum neurofilament light, increasing vertically and looking horizontally at time in years. And then the slopes that are depicted here for the different diseases, Alzheimer's disease, multiple sclerosis, frontal temporal dementia, or ALS, are shown with their respective time frame of when the preclinical disease period is believed to be occurring in terms of years prior to the tipping point of the clinical threshold when clinical symptoms onset and you go from phenoconversion from pre-manifesting to frank disease. The slope is going to be really high actually for NFL. This is a conserved observation across many of these different diseases because the time proximal to phenoconversion tends to have really high levels of neurofilament detected in blood. And so I think it's just important to understand the dynamics here, how, again, neurofilament is not specific for these individual diseases, but if you're using it in combination with clinical diagnostics and features that are relevant to the individual diseases, you can actually use neurofilament as an important marker to tell you important things about what's happening in that specific neurological disease. Next slide, please. Now we're just shifting gears a bit to look at the considerations for NFL in terms of the biomarker validity. It does have fair, and depending on the disease area and the fidelity of the animal model, translational validity. As you can imagine, animal model validity is often a fraught area within neurological disease because these are such complex diseases and modeling complex brain neurodegeneration in a short lifespan rodent is difficult to do, but there is high translational value for NFL within preclinical models. This is just one snapshot of a publication that showed disease progression in mouse models for ALS, Alzheimer's disease, and Gaucher disease, which is a lysosomal storage disorder. The same study did look at a model for Parkinson's disease and did not show progression. So this is not a universal, always silver bullet approach. There is some diversity, but there's of course diversity and variability in the fidelity of the animal models as well. Looking at analytical validity, the assay performance for NFL is robust. It's actually pretty resilient to pre-analytical considerations, and assay standardization has really evolved over the last several years. So I would say that we are certainly at the point where the analytical validity box has been ticked. Clearly, though, you still need to consider data analysis and reporting, and a clear representation of the advancement of analytical validity for NFL is in the very recent breakthrough designation that the Samoa NFL assay was given for relapsing remitting multiple sclerosis. Of course, to pursue this for clinical biomarker development, especially in the context of surrogates, clinical validity needs close attention. So NFL has been shown to have some robust performance as a potential surrogate of disease-related clinical outcomes of interest, but this is variable across different diseases. So the clinical validity is growing, but still variable. And what I'll take you through next is a few examples of where it sits across different neurological diseases and what we can learn from these different applications and diseases that might help us shorten the slope of biomarker development in other diseases. Next slide, please. This is not intended to be a whole encompassing review of the status of NFL in any one of these particular diseases. I just want to drive home the conserved point that it has been robustly shown to have good potential as a differential diagnostic biomarker. So in diseases like Parkinson's disease, as you recall, the animal model fidelity there is somewhat variable. It didn't show NFL in the animal model, but when you look at human NFL levels in Parkinson's disease, it can discriminate atypical Parkinsonian disorders from standard Parkinson's disease, just based on the NFL levels. Looking at the prognostic biomarker potential for NFL, specifically in Huntington's disease, the NFL levels parallel with the clinical evaluation of MRI and pre-manifest Huntington's disease mutation carriers. It also has prognostic potential in genetic inherited versions of Alzheimer's disease. And in looking at a rare disease, spinal cerebellar ataxia 3, which is the most common of the rare spinal cerebellar ataxias, NFL levels correlated with CAG repeat length. So similar to Huntington's disease, the CAG repeat length is associated with earliness of onset and disease severity, and NFL correlated with that repeat length and the worsening of symptoms based on evaluation in comparison to a clinical scale, the SARA score. Next slide, please. This is kind of taking a quick tour through these neurological diseases, going from what would be the lower evidentiary burden and indications of NFL performance as a biomarker, for example, as a differentiation marker and prognostic marker, but then moving further at the end to its potential for prediction. So just closing the loop on prognostic markers for frontal temporal dementia, NFL has been shown to be elevated across all FTD syndromes, in even mild cases, and also in the pre-symptomatic mutation carriers. And these levels increased in the mutation carriers prior to phenoconversion and were also associated as indicators of disease severity. Looking at ALS, again, if you remember that schematic earlier with the iceberg, ALS has a very steep slope. It's a rapidly progressing disease. And evaluation of NFL in ALS has, in concert with other markers, evaluating it with the functional rating scale, has shown prognostic value to determine fast progressors. So this is, again, beginning to get us iteratively into that evidence range of it being able to help differentiate, help stratify, show prognosis, and then if it can help indicate rate of progression, this is beginning to be potentially predictive for the benefit of response to treatment. And that's really what we're looking at in the predictive and prognostic potential for this marker in MS. So it does in multiple sclerosis, it predicts long-term clinical outcomes, and the baseline neurofilament light was sensitive as a predictor to rule out progression into relapsing. And the highest NFL levels were associated with the most rapid progression. So next slide, please. This is just a quick snapshot of a study that looked at various clinical trials for MS, evaluating different outcome measures in addition to neurofilament light. And it showed that when in combination with other outcomes, neurofilament light was helpful in determining progression in multiple sclerosis. This paper, though, did demonstrate that there are certainly challenges to address, including nonspecificity and potential impact of comorbidities. Next slide, please. On this very busy slide, and apologies for the microprint, the takeaway here is that this particular review by Kapoor and colleagues looked at many of the different clinical trials for MS where neurofilament light was an exploratory marker. And the box that I'm highlighting at the red in the bottom is all using the same assay. So now, the trials have migrated to using the Simoa assay and looking at serum NFL levels, and showed that the NFL levels were useful in combination with other clinical outcome markers and provided enhanced sensitivity in being able to interpret changes in response to therapies. So this is, again, building the case that it could potentially have utility for a response marker. Next slide, please. This is actually some of the literature that was used as the foundational evidence for the breakthrough designation of the assay for NFL in MS in order to predict response and risk for relapse. So just briefly, the goal of this study was to develop a control database, because there isn't a normative database that exists for NFL, and then to look at Z-scores, which of course are going to show difference from the mean, and correlate that to the EDA3. This is the evidence of disease activity, and EDA3 means the absence of relapse, the absence of worsening disability, and the lack of MRI evidence of disease activity. So this is a currently not symptomatic MS patient. And they calculated the odds ratio and stratified that across different thresholds of NFL levels. And they showed that the higher the NFL levels were, the odds ratio increased for predicting in these non-symptomatic MS patients the risk of relapse. The arrows at the end of each of these plots just indicate the number of patients above or below the cutoff threshold. And another continued study that they looked at in this particular study was to develop a mixed effects model to look at long-term treatment effects for disease-modifying therapies using this similar approach with Z-scores and NFL levels. They compared untreated groups to platform treatments. This would include first-generation MS therapies like interferon beta, oral therapies that are immunomodulators like fingolimod, that are small molecules that they impact the inflammation cascade, and then also highly effective monoclonal antibodies. And you can see in the first year that the drugs were administered, when they looked at these modeling trajectories for NFL levels, the NFL decreased in every treatment group, but the slope was the greatest in the highly effective monoclonal antibodies. And in the case of the platform treatments, those went back up in terms of NFL score. So this was providing, again, a use case for the potential for NFL to be leveraged as a response marker. Next slide, please. I'm just going to skip over this because I think it's something that has really been reiterated in previous discussions throughout these different sessions. I did want to point out, though, that in addition to the ways that we've discussed how biomarkers can be integrated into the drug development pipeline, the last paper with the modeling work and some of the work that we're doing at the Critical Path Institute also can include biomarkers as a covariate within a clinical trial simulation model that can be submitted through the FDA's Drug Development Tools Fit for Purpose initiative. Next slide, please. So then just wrapping it up here, this is my second to last slide, looking at NFL readiness and evaluating it as a candidate for a surrogate biomarker. Obviously, to show surrogacy, it would need to sensitively track disease progression along with a clinical metric and also capture treatment effects at the mechanistic level. I would say that the maturity of NFL across different neurological diseases is in different places. It's furthest along in MS. Based on existing data, I think we need to look on a disease to disease basis and evaluate what the quantitative link is between NFL and at least one clinically meaningful outcome measure. And then also ask if the NFL is behaving as what confirmatory evidence is lacking. And then as we begin to link it to disease-specific features, we need to ask if it's behaving in a continuous or categorical manner. And then what aspect of the disease continuum is the NFL indicating in these different diseases. So can we reliably anchor these measurements to meaningful metrics of disease such that both are amenable to application in clinical trials? And that is really what we're working on at the Critical Path Institute, because it represents an exciting candidate biomarker, which depending on the disease has utility for prognostic, susceptibility, pharmacodynamics, et cetera. Next slide, please. This is just a summary of some of the challenges associated with developing biomarkers broadly, but neurofilament across neurological diseases, and some of the potential solutions that can address it. I mentioned earlier the lack of normative databases. That's key. We've spoken earlier about the need to develop public-private partnerships and collaborate to share data to really stretch the power of analytics, especially in the rare disease space. And so that's what we're proposing as some of the solutions. And this is what CPATH is working on to evaluate and develop NFL and other candidate biomarkers, along with other clinical outcome assessments and other quantitative drug development tools across our neuroscience consortia, as well as building out infrastructure for non-consortium neurological diseases within our expanded neuroscience program.